What is red biotech? It can be defined as biotechnology developed and applied for medical purposes, diagnosis, prevention, and treatment of various human diseases. Medical application of biotechnology is reminiscent of bloody color, so it is called as red biotechnology. Biotechnology has been developed along with human civilization. Since the middle of 1970s, the genetic engineering and monoclonal antibody production technology have led the revolution of biotechnology that can be readily used in the clinic. Biotechnology can be defined as technology that processes or uses living organisms or substances derived from living organisms for specific purposes. The most primitive form of biotechnology has begun to utilize the natural environment in the form of agriculture and livestock farming. For agriculture and livestock farming in early civilization, selective breeding has been widely used to obtain the crops or livestock of desired properties. As you can appreciate from the fossils found in Mexico remainings, wild corn had been dramatically improved to excellent crop over the centuries by these methods. As fermentation process has been accidentally discovered from food spoilage, fermentation has been widely used for artificial processing of foods and various compounds. Fermentation techniques were performed at the level of cottage industry until the Industrial Revolution. Since then, fermentation technology has grown to industrial scale to obtain various fermentation products such as food additives and antibiotics. As addressed before, development of genetic engineering techniques, monoclonal antibody production, and cell culture technology have enabled us to produce biopharmaceuticals that can overcome the limits of conventional small molecule chemical drugs. Biotechnology can be classified into three categories depending on the scope of application. First, the traditional one is mainly applied for agriculture and livestock farming, or pollution clearance, so-called bioremediation. This is referred as green biotechnology because of the green color of crops. Second category is called as white biotechnology. It involves production of food additives, medicinal compounds, or biofuels, or biodegradable plastics by fermentation. The last one, which is the theme of this lecture, is red biotechnology. As mentioned earlier, developed for diagnosis, prevention, and treatment of various human diseases. The fermentation is the core of the classical biotechnology, so let's take a closer look. Fermentation is defined as metabolic process producing various organic compounds such as organic acid and alcohols by single-cell organisms such as yeast and bacteria. In narrow sense, fermentation is referred to anabolic metabolism of carbohydrates, mainly glucose. Metabolic oxidation of organic compounds such as carbohydrates is called as respiration, while fermentation processes take place in the absence of oxygen as anaerobic metabolism. In general, cellular respiration converts glucose to carbon dioxide and water molecules and generates large amounts of ATP, adenosine triphosphate. However, in the absence of oxygen, glucose can be converted to lactate and only a small amount of ATP can be obtained through the fermentation. Some metabolic products such as lactic acid are essential for growth and survival of the microorganisms. Such metabolites are primary metabolites, while secondary metabolites are referred to microbial metabolites that are not directly involved in growth or survival of microorganisms. The best examples of secondary metabolites are antibiotics, which are produced by numerous microorganisms for suppression of neighboring microorganisms. Industrial fermentation is widely used in 5 areas. First, fermentation can be used for production of the microorganisms as biomass for food sources or yeast strain for baking industry. Second, the microorganism-derived enzymes can be obtained as the final products. Third, a variety of metabolites can be used for dietary or medicinal purposes. And by the microbial fermentation, raw chemicals can be transformed into the desired chemicals. Most of currently used antibiotics are now produced by such biotransformation process. And finally, some microorganisms can be used to clean up contaminated environment. Traditionally, biopharmaceuticals are referred to recombinant proteins produced by genetic engineering, cell culture, and hybridoma cell fusion techniques. However, in recent days, gene therapy using nucleic acid or therapy using stem cells are also included as biopharmaceuticals in a broad sense. Conventional small molecule chemical drugs have relatively low specificity for the target molecules, so the side effects are more frequent. The bioactive proteins and nucleic acid have much higher specificity to the target molecules, so the efficacy of the biopharmaceuticals can be much higher, while the side effects are drastically reduced. A drug is the substance used for treatment or prevention of diseases or injuries. Sometimes another term, medicine, is more specifically used for drug used for the treatment. The natural products derived from plants or animals have been widely used as medicinal drugs for centuries from the beginning of human civilization. In 1897, a chemist Hoffman synthesized aspirin, a pure form of active ingredient included in the bark of the willow tree, which has been known for its analgesic and antipyretic effect. However, the modern pharmaceutical industry, in true sense, has only started from 1930s after successful synthesis and mass production of sulfa antibiotics. Unlike chemical drugs, biological materials were first used for prevention of fatal disease at the end of the 18th century, when Jenner used the vaccinia virus for protection of fatal smallpox. The term vaccination has been coined after the first virus, vaccinia virus, used by Jenner. Biologic products similar to modern biopharmaceuticals have been first used for actual patients in 1920s, when Dr. Banting and Dr. MacLeod isolated the insulin from the pancreas of pigs and used for diabetic patients. In 1950s, many bioactive proteins such as interferons or growth factors have been identified, but the amount of these bioactive proteins are too low for practical uses. This limitation was overcome by monumental development of genetic engineering in 1970s. The genetic engineering, also known as recombinant DNA technology, is one of the core technology for biopharmaceuticals. It involves the separation of DNA coding proteins of interest and insertion of these DNA fragments into specific cells or organisms to amplify the number of genes or produce the proteins of our interest. This recombinant DNA technology can also be used for generation of transgenic organisms by injecting foreign genes in living organisms to have a new trait. For cloning specific gene, the restriction enzymes and ligase enzymes are essential tools required for proper editing of recombinant DNA. Another core technology for biopharmaceuticals is hybridoma technique for production of monoclonal antibodies. In general, one antigen can have multiple antigenic determinants, and each antigenic determinant can bind to different polyclonal antibodies produced by multiple B-cell clones. By selection of single B-cell clone, we can produce very specific antibody with high purity. This monoclonal antibody can be produced by fusion of single clone of B cells producing specific antibody with immortal cancer cells. By doing so, the hybridoma cells can proliferate infinitely like original cancer cells and produce monoclonal antibodies as well. To date, the majority of clinical approved biopharmaceuticals are monoclonal antibodies. These antibodies can be produced in animals, mainly mouse and rabbit. However, the animal-derived antibodies can evoke unwanted immune responses in human body. Such problems can be solved by production of humanized version of monoclonal antibody. Recombinant DNA technology and monoclonal antibody production technology would not be possible if cell culture techniques have not been developed. Cells of multicellular organisms such as human can be taken apart and grown artificially in vitro, in other words, on the glass or in the flask. However, these normal cells, so-called primary cells, can only be maintained for several days or at best several weeks, even in best conditions. When cells undergo rapid aging process after a certain period of time after primary culture, this phenomenon is called a biological clock. Interestingly, this biological clock is not working properly in ever-growing cells such as stem cells and cancer cells. Genetic engineering is the core technology of the red biotech and have huge impact on biopharmaceuticals. The first, genetic engineering technology have solved the source availability problem, especially for the bioactive proteins existing in very low concentration in the body. By simple purification method, these bioactive proteins could not be obtained in enough amount for practical uses. Second, it can dramatically reduce the risk of infection or contamination that can happen during purification or refining step of bioactive proteins from the organisms. Previously, hemophilic patients should get deficient coagulation factors by means of concentrated plasma from hundreds of normal volunteers. However, these blood-derived products were frequently contaminated by fatal viral hepatitis or AIDS viruses. Production of recombinant coagulation factors by genetic engineering can eliminate the risk of blood-borne contamination. Thirdly, we can improve the efficacy of recombinant proteins and at the same time reduce the unwanted side effect by genetic manipulation of protein structure. The best example is recombinant insulin used for lowering blood sugar in diabetic patients. Insulin molecules aggregate with other insulin molecules once they are injected in subcutaneous fat tissue. Deaggregated insulin polymers then dissociate slowly into soluble monomer and circulate the bloodstream before arriving the target tissues. and the insulin molecules are aggregated through specific interaction of protein domains of insulin protein. So, the genetic modification of aggregation domain of insulin can change the degree of aggregation and subsequently determine how fast the insulin can be dissolved into bloodstream. By this method, short-acting or long-acting insulins have been engineered and used for patients. Finally, genetically engineered transgenic animals are a great tool to uncover the action mechanisms of drugs and validate the drug efficacy at the preclinical stage of drug development process. Currently, most of the biopharmaceuticals used in clinics are protein-based drugs. Nucleic acid-based gene therapy and cell therapy are only approved in limited conditions and limited countries. Up to date, clinically available biopharmaceutical drugs include blood clotting factors, thrombolytic agents, hormones, blood cell growth factors, vaccines, monoclonal antibodies, cytokines, enzymes, and more. The development process of biopharmaceuticals is quite similar to that of conventional chemical drugs. In order to develop a new drug, it usually takes about 10 years and more than $1 billion US. Most of this time and money are used to verify the efficacy of the drugs in actual patients via clinical trials. The clinical trials are divided into 3 phases according to the purposes and study protocol. The phase 1 clinical trial enrolls 20 to 100 healthy volunteers to test the short-term side effects and determine the maximum tolerable dose for patients. In phase 2 trial, about 100 patients and normal controls are tested for the relatively short-term toxicity and efficacy of the drugs. The final step, the phase 3 trial, large numbers of patients are tested for long-term side effects and efficacy. Only the drugs that survive that all 3 steps are ready for clinical uses after approval by FDA. Even though the drug development takes a lot of time and money, many multinational companies are eager to develop new drugs. Then why? It is because the drug industry is well-known high-risk, high-return market. Drugs are sometimes called a blockbuster if the annual revenue is more than $1 billion US. Imagine that you own several such blockbuster drugs, then your revenue is guaranteed for more than 10 years by patent, which will eventually exceed the money you have to pay for drug development. Biotechnology can be patented if the technology satisfies following 4 criteria, which are novelty, non-obviousness, sufficiency of disclosure, and utility. Patent is possible if the hand of man has played an obvious part in developing the product. By this principle, many products of nature have been successfully patented by enriching, purifying, or modifying natural products such as specific antibiotics, microorganisms, and proteins. However, as biotechnology makes enormous innovation, there have been arising ethical and political issues and legal patenting, such as human cloning and using human embryos for commercial purposes. Summary of this lecture is as follows. First, biotechnology refers to technology processing life itself or substance derived from live organisms for specific purposes. Depending on the scope, it can be further divided into green, white, and red biotechnology. Second, the red biotechnology is used for disease diagnosis, prevention, and treatment by using genetic engineering, cell culture, monoclonal antibody production, and stem cell technology. Third, the biopharmaceuticals are proteins or nucleic acid and have higher specificity against drug targets compared to conventional small molecule chemical drugs. Therefore, they have higher efficacy and less side effect. Fourth, genetic engineering technology dramatically eliminated the risk of infection or contamination. In addition, it can improve the efficacy or reduce the side effects by changing the protein structure of original bioactive proteins. The fifth, development of biopharmaceuticals is similar to that of conventional drugs, which includes preclinical trials using laboratory animals and clinical trials. Sixth, biotechnology can be patented if they satisfy following four criteria: novelty, non-obviousness, sufficiency of disclosure, and utility.